Hi, biology. This is Miss Satterley. I'm going to go over the PowerPoint that was posted yesterday on the scientific method. If it'll let me. Okay. So the scientific method is a process that scientists use for experimentation. There are different ways that you can actually set up the scientific method, but we're going to go with this one. So in our example, we have 6 stages. First one is observation, and then hypothesizing, predicting, experimentation, analysis, and then producing a model. So step 1 is observation. Observation is the act of noting or perceiving objects or events using one or more of the 5 senses. This is always the first step to the scientific method. In the 5 senses, you can use sight, smell, hearing, taste, or touch to collect observations. Step 2, you then take those observations and you form a hypothesis based on them. A hypothesis is a proposed explanation that might be true, and it's based on those observations. The rules for making a hypothesis, though, it must be testable and it needs to be able to either be supported or disproven. Step 3 is predicting. A prediction is what you expect to happen if your hypothesis is correct. So you make your hypothesis first, and then you will make a prediction based on it. Every time a prediction is usually when a prediction is written, it's actually written in an if-then statement. So if this happens, then this will be the result. So that's how a prediction is set up. Step 4 is experimentation. So this is actually testing your hypothesis, trying to see if it is supported or disproven. Experiment is the process of testing a hypothesis or prediction by gathering data under controlled conditions. And later on in the PowerPoint, I'll talk about what a controlled experiment is. Another way to define experiment is a series of steps used to test a hypothesis. Step 5 is to analyze. So after you collect your data from your experiment, then you go through an analysis. An analysis is a process of determining whether data is reliable and whether it supported or did not support your hypothesis. Step 6 is to produce a model. A model is a representation of an object, system, or process that shows a relationship. Models can be visual, verbal, or mathematical. So this is how you present your data after you have done your experimentation and analysis. Data collection. So what data is? Data includes any and all information that scientists gather during experiments or observations. Data collection can include observing, measuring, sampling, or organizing data. The first one is measuring data. There are 2 ways to measure data. You can either measure data in a quantitative way or a qualitative way. Quantitative data is data that is counted or measured in numbers. Qualitative data is data that is descriptive. So this includes color, texture, shape, or anything that's going to describe something without using numbers. Next one is sampling. Sampling is a technique of using a sample or a small part to represent an entire population. Sampling is only useful, though, if it has a large and random sample is taken. If you have a sample that is not random and not large, it can actually skew your results and make your whole population seem different than it actually is. Next one is organizing data. Data is useless when it's unorganized. So you can't read it, you can't understand it, so you have to organize your data in order to make a logical analysis of it. The ways to organize data include charts, graphs, tables, and even maps. Ways to analyze data. Statistics is a way that scientists can analyze data. This is a technique of determining relationships between variables. Statistics are useful because you can compare your data with other studies that tested similar things. It also helps determine sources of experimental error. An experimental error is going to show you how accurate your experimental results are. Okay, let's talk about what experimentation actually consists of. Most experiments need to be a controlled experiment. Controlled experiments are experiments based on a comparison of a control group with an experimental group. A control group is going to be a group that is not going to have any kind of what's called an independent variable added to it. So this picture down here, I'm going to talk about this a little bit more tomorrow with just a short video, but you have a control group and an experimental group. The control group and the experimental group are going to have something in common. One, they're going to have the same size pots, they're going to have the same type of soil, and they're going to grow the same type of plant. Now down below the experimental groups, you can see that there are things added to those plants. Water is one of them. Water is a control, which means it's also added to the control group. Now, what's being added that's different is our types of salts that are added. So there's a nitrogen salt, a potassium salt, and phosphorus salt. And that's what we call our independent variable. An independent variable is what is manipulated or added to an experiment. And you're going to measure plant height, which is your dependent variable. Again, I'll discuss that all a little bit more tomorrow with just a short video. Okay, a control group. A control group is a part of an experiment in which the key factor is not changed. It's used in comparison with the experimental group or experimental results. So you're going to do everything to your control group that you do to your experimental group without adding your independent variable. Now, the experimental group is the part of the experiment where you add the independent variable and you manipulate it or change it. These two groups are identical except for this independent variable. Now, a variable is any factor that can be controlled in an experiment. There are three types of variables: independent, dependent, and a controlled variable. So in this picture here, we just have a brief definition of each one. So independent variable is what you change. So N is in red, meaning that's what you put into the experiment. Dependent variable is what you observe. So that's the results of the experiment. So what happens to each group after you add the independent variable. Now, your controlled variable is what is kept the same, and that is to make sure there's no other factors affecting your experiment except for your independent variable. Now, the independent variable, again, is a variable that is manipulated or changed during an experiment. If you make a graph of your data, you're actually going to put your independent variable on the x-axis. Now, your dependent variable in the same graph is going to go on your y-axis. A dependent variable is the variable that is observed or measured during an experiment. So this is the data that you're collecting as your experiment goes on. Your controlled variable is a variable that is held constant. So the example that we had before, our controlled variable would be the amount of water and the type of soil or the type of plant being used. Now, experimental support. The whole point of an experiment is to support or reject a hypothesis based on the data. So if data does not support, you're going to reject your hypothesis. If data does support, it's not rejected. A hypothesis is never going to be completely proven because there can one day be an experiment that causes that hypothesis to not be true. It can just be supported or rejected. There's a couple other vocabulary words that I want to go over. First one is scientific theory. A scientific theory is a unifying explanation for a broad range of observations believed to be true. There's a lot of different theories in science, especially in biology. Some theories that we'll talk about this year include cell theory, the theory of evolution. The next one is the scientific law. Scientific law is related to scientific theory, except a scientific law is a statement that summarizes a pattern found in nature. Now, a scientific theory, I want to go back to that. This is something that's actually supported over and over. Multiple experiments have been done. They all have the same results, and that's how you get a theory. Something that is supported, it has a testable explanation, and a hypothesis over and over. So that's a theory. Now, scientific law summarizes a pattern in nature. So gravity is 9.81 meters per second squared. That is what gravity is on Earth. That is a scientific law. It does not change. Now, if you went to the moon, gravity is 1.66. But that is a different scientific law because it's a different body in space. So that's what scientific laws are. There are the laws of motion in physics that are also laws as well. Last one is an inference. So this is the last slide. An inference is a conclusion made on basis of facts or premises rather than direct observation. So you can make an inference after looking at some data, and not data that you maybe have done experimentally, but different facts that you find or different data that you got from somewhere else. And you can make an inference. The thing with an inference, though, it is not testable. It is what you think and what you know, and then you infer. That is the end of the lesson, so it's a little shorter for today. Please make sure you actually watch this video. Make sure you have your notes. There's a homework assignment posted. Some of you, I think, have already started to work on it. But make sure you don't try to do that without actually having your notes in front of you. On Thursday, there is an assignment that will open up. It's called Scientific Method Analysis or Application Questions. It's going to include some different examples of experiments. And I'm actually going to do a short video on that and post it tomorrow, so you guys can look at that while you do that assignment. That assignment is actually not going to be due until Monday. I'm going to give you a couple days to work on it. Make sure you don't get frustrated with it. It is going to require a little bit of critical thinking, but there are questions that have more than one correct answer. All right, that is it for today. I hope you guys have a good day, and goodbye.